We know that we can perceive these blobs, and we perceive blobs that are placed into different contexts because of what we're inferring based on the context. So, what's going on here? What role does inferring play in perception? Well, um, obviously there's been a lot of work in this. So, the first idea the first scientific idea on the role of inference in perception was proposed by Helmholtz, who talked about this theory of unconscious inference, right? This idea that we do, in fact, use unconscious processes to infer what stimuli are based on their context, and that stimuli can be perceived as many different things based on the context, right? Um, Part of the theory of unconscious inference is the likelihood principle that objects are going to be perceived based on what is most likely to have caused the pattern. Again, we're going back to context, right? So we're looking at the context of the of how this object is appearing within a scene, and we are calculating, we're unconsciously inferring what is the most likely thing that this object is going to be based on its context, right? Um, for nowadays, we have actually applied different statistical techniques to try to actually model this sort of inference, and one of the big techniques that's used nowadays is what's called Bayesian inference. Essentially, what Bayesian inference is calculating probabilities of what something is based on evidence, right? So the more evidence that you gather, you change the probability as to what something might be. So, for example, with Bayesian filters are used to filter out your email, right? To figure out what is spam and what is not spam. And the way these filters work is that they parse every email and they gather a bunch of evidence. So they look for certain words in the email, like Viagra and things like that. Look for certain addresses, and they add up all of these different pieces of evidence to come up with a probability that yes, this email is spam or that it's good email. Um, Here's an example of likelihood and inference, right? So when we're looking at the pictures in A, right, we're seeing what we perceive as two rectangles, a red one and a blue one, and the blue one is overlapping the red one. And if you were to alter the angle a little bit that you're looking at in angle B, this is what we would infer we're looking at, right? We infer we're looking at two complete triangles, but it could in fact be C, right? We could actually be looking at this, right? And the reason we typically don't infer C, but we do infer B, is because it's more likely, right, that it's going to be B. We encounter more. We're using the fact that these two are similar to each other, so we're grouping them together, the similarity, right, similarity principle, and so we're assuming that when we're inferring that when we're perceiving these two objects, that the one behind it is a complete rectangle, but it doesn't have to be, right? I mean, it's could be a mistake of our inference that in fact what we're seeing is this here. Um, Another experiment that was done to link, or an experiment that was done to link neural activity in the brain to object perception, right, was done by Grill and Spector, right? And what they did was they looked at the fusiform face area. This is the part of the brain that becomes activated when we're perceiving a human face, okay? And so here's what they did, right? So they did a trial, right? And they showed these pictures very, very quickly, like for 50 milliseconds, right? So people would either see a picture of Harrison Ford's face, the actor from Star Wars and Indiana Jones, right? Either a picture of his face, some other person's face, or just some random pattern, okay? So they would see one of those three things and then the random mask, right? So basically, then they were asked, what did you see? Okay. Now, and at the same time, the activity in their fusiform face area was measured. And so they had 60 pictures of like this that were presented, one right after the other. So they would flash it for 50 milliseconds, the random mask, and then they would say, okay, what did you see, right? Um, they found that the fusiform face area only responded to faces, right? So whenever the subjects perceived a face, right? Whenever the subjects were presented with a face, right? The fusiform face area would become activated. It wouldn't become activated with the random pattern, but it would become activated preferentially to the faces, right? Um, the other interesting thing was that the highest activation occurred when the subjects correctly identified Harrison Ford, right? So he was the only person whose face they recognized. So one of the assumptions of this is that when you recognize a face that from your past, the fusiform face area is most active, right? And sure enough, whenever for that 50 milliseconds when the face was presented, and then they were asked what they saw in that 50 milliseconds, uh, and if they said Harrison Ford, it turns out that their fusiform face area, their FFA, was lit up the most, right? Um, and this demonstrates to us that the neural processing, right, not is related to not only when the stimulus is presented, right? So when it gets presented, the FFA lights up, but also to the response, because the correct response to Harrison Ford was correlated with highest activity, right? So remember that highest activity happened when the face was presented, and then it went away. And while it was gone, the subjects were asked, "What did you see?" Right? And whatever happened in that moment, in that 50 milliseconds during the highest activity, influenced the perception, what was seen. Okay, which is pretty interesting. So here's an example of the experiment, right? So we flash for 50 milliseconds Harrison, and then you get a 50 millisecond mask, and then we say, "What did you see?" Right? Did you see Harrison? Did you see something else, or nothing? Right? Um, and that's the way the experiment was done. And remember that, remember that if they identified Harrison correctly, right, it was associated with a much higher reading, a much higher signal in the fusiform face area, right, in the time immediately um, uh, when the signal was presented. Another experiment, Scheinberg and Logothetis, I can't say that right. So they had this monkey, right, that was trained to pull a lever, right? So whenever the monkey was trained to pull the lever whenever it saw a sunburst, or another lever whenever it saw a butterfly. So one lever for sunburst and one lever for butterfly. And then they used this technique called binocular rivalry, where they basically put these goggles on the monkey and they showed a sunburst in one of the eyes and a butterfly in the other at the same time, right? Butterfly and sunburst at the same time, right? And then what they did was they monitored a neuron in the cortex that had previously only lit up, that had only been activated whenever the monkey perceived a butterfly, right? And what happened was anytime here's the monkey seeing both at the same time, right? Seeing the sunburst pattern and the butterfly. It's at the same time, right? So it's on all the time. It's at the same time, right? So whenever the monkey is perceiving the sunburst, so if he focuses on this eye and he decides I'm gonna look at the sunburst, I'm gonna focus on the sunburst, um, the neuron wouldn't fire, right? The neuron wouldn't fire, but he's pulling on the sunburst lever, right? He's pulling on that lever to get his food. And then if he all of a sudden starts pulling on the other lever to indicate he's focusing on the butterfly, the neuron that only responds when the butterfly is perceived starts firing, right? So this indicates a very interesting correlation between what we think is going on as a perception, an internal perception being represented as this neuron firing, right, and an actual behavior that is telling us which object this monkey is perceiving. Right. It's really interesting because the monkey is focusing its attention back and forth between the sunburst and the butterfly because they are both being presented at the same time. Right. Another experiment by Tong, he used the same technique, binocular rivalry, right? Showing basically two things at the same time. And basically what he did was use like this red-green filter, right, where they had these goggles that would, one eye could only see what was colored red and the other eye could only see what was colored green. And then they showed this picture with a house and a face superimposed on each other. The house was red and the face was green, right? So house was going to one eye, face was going to the other eye, right? And basically the folks were asked, push a button to indicate what you're looking at. Are you looking at a face? Or are you looking at a house, right? And they're both being presented to the eyes at the same time, just like the monkey, right? Just like the monkey. And then they measured MRI activity in two different places. The parahippocampal place area, which lights up whenever we're perceiving locations in space, and then again the fusiform face area, which lights up when we're perceiving faces. And sure enough, whenever the person was pushing the button to say, yes, I see a face, as you see here in this experimental setup, the fusiform face area would light up. And similarly, whenever they were pushing a button to indicate that they were seeing the house, the red one, the parahippocampal place area would light up. So again, we have this really neat demonstration of our ability to focus our attention from object to object and to correlate this focusing of perception from object to object with different parts of the brain that allow us to perceive these things. Reading, right? Reading is a specialized thing that we humans do, right, with our perceptions, right? So one of the things that we want to try to understand are what are some of the mechanisms that are going on with reading. An experiment by Kamitani and Tong helped us to understand this. And basically what they did was they looked at the way that a part of the visual cortex responded to different gratings, right? So remember, the gratings are those lines that can be oriented either horizontally or vertically or at an angle, right? And so they presented gratings of different orientations to people, and then they recorded the activity in a little section of cortex. And whenever you do these MRI studies, the sections of cortex that are being, or the sections of the brain that are being studied, are broken up into little boxes that are called voxels, right? So the resolution of the MRI is not down to the neural layer at all. It's really, these little boxes that are a few millimeters across. That are called voxels, right? So we looked at the voxels that were associated with a part of the visual cortex, right? And we looked and saw which voxels lit up with which orientations, right? So whenever we were perceiving this orientation, these voxels would light up, this orientation, et cetera, right? And then we said, okay, we've looked at the patterns of how the voxels light up in the brain based on the orientations. Can we then attach the readout, or can we monitor this activity in the brain with these voxels and then predict what it is the person is looking at? So in other words, can I interpret the output of the voxels and figure out just from that what the person is looking at? And sure enough, they can. They were able to use a decoder to look at the pattern of responses in the brain and these voxels, and they were able to correlate different patterns with what the person was actually looking at, right? So here we have this example, right? This person's looking at these different orientations of gratings, and it goes into our visual system, goes into the back of our brain, into our visual cortex, and from there, these little voxels all light up in a certain pattern.